First of all, thank you very much for the LLB host and Divya Shri, and also TNAU, CPMB, and the organizers, everybody, for having called me for this talk. It's really a great pleasure to address you people, and especially because I'm associated with TNAU, it's a matter of pride to involve myself with something with TNAU. And firstly, I want to tell you that I'm a plant physiologist basically. I'm not a virologist or a drug discovery or something like that. But the whole study is about. It started off with a small intuitive study. It was like, if you remember, the whole thing started. The outbreak started somewhere around January 15th, and we work on NICRA. There's a NICRA project of ICR. I am a PI of a project where we do computational genome analysis and gene mining in maize. Basically, we try to find out genes and candidate genes for drought tolerance and other abiotic stresses, heat, drought tolerance, like that. And that is the project I work on. And my project staff, that is a bioinformatician, also works on this. So what we did was, what we thought of was, when the genome of this virus started to come in NCBI, we just thought that why not we do some practice on analyzing this genome and seeing how it is related to the other viruses, known viruses and sequences and all that. It was just a thought that struck my mind, and I asked my project staff, Divya, Banu, to download the genomes and start doing some analysis. And that was how it started off. That then, another thing I'll tell you is, the whole study is a very simple study. There is nothing very special in it. We use very, very simple bioinformatics approaches, very accessible and open access server-based analysis. We did everything from homology modeling to protein ligand interactions, and also docking studies. Everything was server-based and very not computationally very intensive. It was all simple things. Only thing is, we started off very early, in the sense that there were only 6 genomes of the virus available when we started off. And also, there was no knowledge about the basis of the virus. The virus was not even named at that time. In fact, we didn't know that it was related to the earlier SARS. And that was when we stuck up our pay dirt. Like, we saw that most of the homology modeling led us to proteins which were clearly related to the earlier SARS. And immediately what we did was, we put it up in a preprint server. So people who are listening to this lecture, and the younger people, like younger researchers, I would like to tell you that preprint servers are not very popular with biological researchers. It's very popular with physical sciences. But here, let me tell you that if you find something interesting and find something new and novel, I would suggest that you put it up in a preprint server, so that you can maintain your priority. That is, prior art is maintained. So that is what we did. Simple study it is. So now I'll get into the study as such, proper study. This is actually, like you can see in the title, it's a whole genome analysis of this virus, SARS-CoV-2. And we did some homology modeling of the main protease and non-structural protein 3. And we found out 2 compounds, that is an azapeptide and a lead inhibitor with possible antiviral properties. And our team is Divya, Banu, Anjani Aluri, and Samriddhi Gupta. All of us from different places. Actually, Divya Banu is my project staff, Anjani is my student, and Samriddhi Gupta is from the School of Life Sciences, Department of Biochemistry in University of Hyderabad. So what we did was, and we actually today, as a matter of fact, I would like to share that today is when our final paper has come online in New Journal of Chemistry. This is the publication. It has come out today. I'll share the URL with you if you want the full text. Now, here is the hypothesis. Actually, what we did was, we actually hypothesized that this virus can have homology with several proteins of the earlier SARS. We thought that there must be definitely a high degree of homology. So we saw that these proteins, the PL-PRO, that is a papain-like protease, and 3CL-PRO, that is 3CL-like protease. These have a very important function of processing the viral polyprotein. Polyprotein is a large protein of the virus, which is further cleaved into several other NSPs, or non-structural proteins. So this was the basis of our starting off with the study. That is, the hypothesis was that. And this PL-PRO is to be noted is that it performs a function of stripping the ubiquitin in the host. That is, it facilitates the virus to replicate, and it also facilitates the virus to evade the immune response. This is what I want you to note. It helps the virus evade the immune response from the host. So that is a very important protein. That is a very important target for drugs, because once we target that successfully, we can not only induce immunoprotectant in the host, we can also stop the viral replication. So it's a dual sort of approach if we go for PL-Pro. And they also have other roles of disrupting signal cascades in infecting cells and protecting the uninfected cells. So these two proteins, which form the main protease, which are in the main protease, are very good targets. So with this hypothesis, we started off. And actually, to go into, I think this probably most of you would know, must have read about the coronavirus. It is basically a zoonotic. It has got zoonotic properties. It jumps from species to species. It has been jumping from bats to humans, camels to humans. The SARS, which was earlier found, was 2002 in Guangdong province. That was the first of this. Although there are more coronaviruses that actually affect the humans, there are 7 in fact. These 3 form the main ones. That is the SARS, the MERS, and the present one, that is the SARS-CoV-2, that is taken a pandemic form now. Now, now going to the study, Ash, I would want to just briefly tell you about the genome structure of the virus. The virus is, actually has a 27 to about 30 kilobase genome size. It is quite a large size for a virus. Most viruses don't have this size. This is quite a large size. So, and they are especially this viruses, like coronavirus in general, are positive sense RNA viruses. That is, it is already a processed mRNA. Once it gets into the cell, host cell, it directly starts to transcribe. That the protein starts to transcribe. There is no DNA to RNA, or there is no reverse transcriptase like we have in HIV and all that. So this is a very adapted advantage for the virus. It is a positive sense RNA. It gets into the host and immediately it starts its work. And this has 2 main ORFs. That is one ORF 1A and one ORF 1B. There are several other ORFs, but these are the main ORFs. And these ORFs actually correspond for polyproteins that are processed, and 16 non-structural proteins are processed from these polyproteins. And the remaining portion has got a structural protein. That is, these are the non-structural proteins. That is 16 non-structural proteins. And they have, it also has a structural protein, which is a spike protein. You know the spikes that you must have seen in the photographs or pictures of this virus, the spikes that jut out of the virus. That is a spike protein. There is an envelope protein, there is a membrane protein, and a nuclear protein. This is the basic structure in nutshell of this virus. And it is also known that this can undergo, one important thing of this virus is it can undergo recombination and in the genome within the host cells, and it can recombine with other coronaviruses. That is how probably it evolved. The origin is actually a big question mark. We don't know how actually it came. We don't know the intermediate animal also. So, but it has got a recombination factor, that it has got a high rate of recombination. And this is one of the important aspects by which it adapts itself to new hosts. That is the new receptor cells and all that. So these factors give it a very high adaptive advantage and the capability to jump across species. Now, this is the genome structure. Let me tell you, like I told you before, this has got a about 30,000 BPS genome structure, and the ORFs, like I mentioned, ORF 1A, 1B, and several ORFs of ORF 2 to ORF 10. 1A and 1B are important for the non-structural proteins, which we'll be discussing. So this 1A and 1B almost forms the about 20,000 of the base pairs. The rest of it is, this 10,000 forms the rest of the base ORFs, which is 8 ORFs. So in this is actually, I have taken this genome structure from a study where the authors have actually done discovery of potential drugs by computational methods. That correctly corresponds to what study I have done. And these things that is mentioned here, that is these are all the PDB entries of the particular protein. Like for example, it is NSP5, the main protein which I talked about. NSP5 and NSP3 is where we dealt with. NSP5 is the main protein. This is the PDB entry of NSP5, and NSP3 is over here. NSP3 is here, and the PDB entry is 36A. So these two proteins is where we targeted. So the rest of the proteins, like I told you in the previous slide, the spike protein, the envelope protein, the membrane protein, and the nuclear protein. Here, now coming to the main aspect of our study, like how did we go about and what did we want to do? To tell you the truth, we didn't go on starting off to find an antiviral or a vaccine or something like that. We just wanted to analyze the genome. We wanted to see if there is a homology with other viruses. We wanted to see what this virus was. That was the basic objective. That was the basic intuition behind the study. But when we kept going, we saw that the NSP3, that is non-structural protein 3, I'm sorry, one second. Yeah, this non-structural protein 3 here, this thing, and the CL3 Pro, both were having a very high degree of homology with the previous SARS. That is where when we did online homology, we saw that about 50 templates matched with them, and the top templates were exactly almost very highly identical, 96%, 98%, like that with the previous SARS. So at that time, the virus was not named SARS-2. And the papers were coming out. This was about February 4th or something like that. February 4th it was. So papers were coming out in bioarchives and chem-archives. And people started to speculate on how this virus is related to the previous SARS. That is when we put up our paper. And then we put up our paper saying that the CL Pro and PL Pro were closely related to the earlier SARS. So this is how we went about. And to know, tell you about these two proteins, that is NSP3 and NSP5. They are involved in the processing of viral polyproteins, like I told you before. And they actually split the viral protein into several NSPs and package it into new generation viruses. This is the main function. So this is why these two proteins can be very good targets for drug development. Then the computational analysis we did was basically we started off with the homology modeling. Like I told you, we did it with known templates. We did it with Swiss model servers. That is a very automated sort of homology modeling, not much of a manual modeler and all. We did not use that. We just uploaded our sequences, and we did a homology model, and we got back query results from Swiss models from several templates. We started analyzing the templates for similarity and also identity. And we saw several templates. Like I said, we saw several templates. We just, when we looked through the template, that is when we saw that certain templates, that is one or two templates, had ligand complexes. That is where we narrowed down. We omitted the templates which did not have ligand complexes. That is, most of the templates already in the PDB database were just the crystal structure of the earlier SARS. Earlier SARS had been worked very well, and several other coronavirus had been worked very well. So PDB had a lot of entries of similarity with this protein. But what we did was we narrowed down on homology models, templates which had ligands with them. And some of the ligands were not actually antiviral ligands. So we further narrowed down to ligands from literature which had antiviral properties. So that is what we thought, that the viral replication, that is, we further narrowed down to ligands which could possibly stop viral replication by inhibiting these proteins. So that was the idea. And this is where this is the...
Hello, sir. Sorry for the interruption. I just want to know why the filtration criteria has, the structure has ligands. Why you kept as a filtration criteria?
Actually, why? Because when we started to get the templates, that is when we thought that we can look for ligands which can have antiviral properties. Otherwise, getting a template without just a template which has similarity with the SARS, we only know the tertiary structure of the M protein or PL Pro. That is already known. So that wouldn't form a discovery or a practical applicability. So when we saw that there are templates with ligands and those ligands had antiviral properties, that is when we thought that this could have practical applicability. That is where we narrowed down to those ligands.
Okay, thank you, sir.
This is the pictorial description of how we did it, actually a graphical description. That is the two candidates we got, GRL 0617 and AZP, that is azapeptides. And the targets were these. Like you can see, we narrowed down to the amino acid sequences of the targets. That is between 315 amino acid in the frame 2 and a 306 amino acid in the frame 2 between these residues. So here, So here we saw that one had 82% similarity with PL Pro, and that had a ligand GRL 0617, and the other had 96% similarity with C3L Pro, and that was AZP. This is how we narrowed down on the two particular antiviral targets. I'm sorry, antiviral drugs. Now we did a close alignment with the Clustal, and here you can see this is the query sequence. This is the query sequence. This is the template. Same the other model again. This is the query sequence. This is the template. There was a 1-to-1 match. That is almost every amino acid was matched in this particular area. That is, there was a 306 amino acid match in the C3L Pro and a 317 amino acid match in the PL Pro protein. Now, what we did was we took it as a starting point for the drug design. Actually, we didn't actually design drug design de novo. We did not do de novo drug designing. We just matched it with the earlier literature. So here, then what we did was, now this, we had to see if there is a protein ligand interaction with our model as we could see in the template. This is the model we developed. Here is the ligand attached to the C3L Pro. C3L Pro, that is of the main protease. This is the azapeptide. This is our model, which we got from the template. Similarly, this is the other model, PL Pro. This is the GRL 0617 ligand attached to the homology model. Now here, the next step we did was we wanted to compare the binding sites of the model and the template. So here on the right side, this is our model from the SARS-CoV-2. This is the template. We saw in this thing that is a 3E9S PDB template, which binds to GRL 0617. We saw that almost every amino acid in the binding region matched, except that there was a specific tyrosine here. You can see a tyrosine that was an extra tyrosine in our model. So we had to go further ahead for with Autodoc Vina. Similarly, in the other azapeptide, that is a 2A51, there was a complete 100% match. You must have noted that this azapeptide had a 96% identity with the earlier SARS, whereas the earlier one, that is GRL Pro, had only 82%. This azapeptide had about 12 amino acid differences. So here we did an interaction profile with the protein ligand interaction servers.
Hello, sir. Sorry for the interruption. There is someone who want to ask question. So can we allow him ask a question?
Please do. Please do. I'll try my best to answer.
Yeah. Hello, Mr. Lakshman Kumar. Can you ask a question? Hello. Hello, Mr. Lakshman. Okay, I think he's not there. Maybe we can continue like this.
Yeah.
Sorry, sir.
Now, yeah, I think he's come or what?
He's not responding, but he.
Okay, then I'll continue.
Okay.
So
Okay.
what we did was next we thought the next step was to find out how well this model or how well these antiviral compounds ligands GRL 061 and 0617 and azapeptide will bind with our homology model. Theoretical chemistry basically it is. So we went into yeah, Lakshman Kumar has raised a hand. Probably he wants to ask a question now. Stop.
Seems like okay.
I saw
Hello, Mr. Lakshman Kumar. Do you have any questions? Okay, I think it might be a mistake, I guess.
Okay, then I'll carry on. I'll continue with my presentation.
Okay.
So
Okay,
what
sir.
we did was we went into a protein ligand interaction servers and we did PLIP database. We collected the PLIP data so as to find out the hydrogen bond, hydrophobics, and alkyls, pi and interaction, pi sigma interactions and all. I'm actually not very familiar with this, but my other collaborator, Samriddhi Gupta, had done all this work. She did that in Autodoc Vina, where about 9 confirmations of this ligand were run in the Autodoc Vina, and we found out which one of the confirmation had the maximum affinity. The affinity of about -7 is an accepted affinity for a good ligand binding. So this we did. You can see that in this GRL 0617 with CT PL Pro, there was one which had unfavorable donor, whereas there was a good affinity. Although I must say that in comparison with the earlier SARS, that is reported in literature, and our homology model, there was quite a bit of a difference in affinity. That is the reported one in literature with the earlier SARS had an affinity of -10 or minus 10 point something, but we only got about -7.1. -7.1 is, although I must say theoretically, a good affinity and a good possibility of good binding. So here is the next one. The other compound, that is the interaction profile of AZP. AZP had a much better binding affinity than the GRL1 6017. So here I'll just conclude in a small nutshell. That is basically we had a template to a we had this template from ADB which had 96% identity with the C3-like peptidase, and the compound was azapeptide epoxide, and this had a binding affinity of 7.1. Similarly, the 3E9S of the PDB database of the earlier SARS had 82% identity, and the compound was GRL 0617, which had a higher, much better affinity. Although, it must be of -7.9 kilocals per mole. To note here was this, which has a lower affinity, had much higher identity in terms of the alignment, that it had a 96%, whereas this one had 82%. So another thing later on, we kept revising our work. As more and more genomes were being sequenced and deposited in NCBI, we did a whole analysis of about 831 genomes of this virus and found that this particular area, that is 306 amino acid sequence and 315 amino acid sequence, was totally conserved. So we can very much say that there is no, this 813 was done on April 24th. That is almost about 2 weeks back. So it can also be said that there's not been any much mutations in this virus. If you have been reading literature, you would probably have noted that there are mutations in this virus which can change the protein structure, like tertiary structure can be changed and binding can be changed because of mutations. We saw that this was not the case. That is these two proteins, that is C3L Pro and PL Pro, and the region, specific region of that is a 315 and 306 amino acid region of that, did not have much of mutations, and they were conserved in 831, and I think probably they are well conserved proteins. And both had, this is what I said, and one of the things from the literature, that these two compounds don't have cytotoxicity to humans. So targeting this part of the genome for antiviral compounds can have a good implication for the development of antiviral compounds for SARS-CoV-2, and it is possible that these ligands we found can be used as antivirals for SARS-CoV-2. And before finishing off, I'd like to tell you from where we got this. That is from our homology models. The main templates we narrowed in on was from the azapeptide was from this paper. It was published in 2005, and a substrate like azapeptide, which inhibited coronavirus. This is a laboratory work done, main peptidase being inhibited by azapeptide. And the next paper is this one, a non-covalent class papain-like protease inhibitors. That is a GRL ligand. So these two are the main papers from which we drew our templates. This is as much as I can do. I think you must have had some good information from this. Thank you very much.